In the last video, we explored how muscles contract using the sliding filament mechanism, and I hinted at an important question. How does the electrical signal from your nervous system actually trigger a muscle to contract in the first place? The answer lies in a specialized communication site between a motor neuron and a muscle fiber called the neuromuscular junction. In today's video, we're going to walk through how the nervous system talks to the muscular system step by step, so you'll see exactly how the signal gets sent from the neuron to the muscle to allow for muscle contraction to happen. We will see how a thought like "pick up my water bottle" turns into an action. To move your body, your brain has to send a message to your skeletal muscles. But here's the catch. Neurons and muscle fibers don't directly fuse together. Instead, they communicate across a very tiny gap using chemical messengers. This gap is called the neuromuscular junction or neuromuscular synapse. When you hear the term synapse, you should immediately think that this is where a neuron meets another neuron, a neuron meets a target cell, or a neuron meets the muscle. So a neuromuscular synapse is specifically telling you that this is the place where a neuron and a skeletal muscle meet. At the synapse, there is a small gap called the synaptic cleft, where the neuron can send signals to its target, another neuron, a muscle cell, even a gland. These cells do not physically touch, so the first cell, the neuron, will communicate with the second cell by releasing chemical messengers called neurotransmitters. We will talk more about how neurons interact with other neurons and target cells later on when we get to the nervous system. For now, we are going to focus on the neuromuscular synapse, where a neuron meets a skeletal muscle cell. Let's take a look at the neuromuscular junction and the main characters we find there. First, we have the motor neuron. This is the nerve cell that carries the action potential down its axon. It is going to go down to the very end of the neuron, which is the axon terminal. This part of the neuron sits very close to the muscle fiber and divides into a cluster of synaptic end bulbs. These synaptic end bulbs, or boutons, contain synaptic vesicles, which are membrane-enclosed sacs that have neurotransmitters inside them. Here, we find that the synaptic vesicles contain acetylcholine. Acetylcholine is the neurotransmitter or chemical messenger that is going to actually carry the signal across the synaptic cleft. The synaptic cleft is the tiny space between the neuron and the muscle fiber. This again is where acetylcholine, that signal, is going to travel from the neuron to the sarcolemma, the muscle's cell membrane. The motor end plate is going to be that specialized region of the muscle cell's membrane that is packed with acetylcholine receptors. So the neuromuscular junction is the meeting place where the neuron will send its chemical signal, which is the neurotransmitter, specifically in this case acetylcholine, into the synaptic cleft to go towards the muscle. Now that we know what the neuromuscular junction looks like, let's take a look at how the nerve action potential signals a muscle action potential. A nerve impulse is going to travel down that motor neuron and reach the axon terminal. Here, voltage-gated calcium channels in the terminal are going to open and calcium rushes into the neuron. These channels open in response to the voltage change from the action potential moving through the neuron's membrane. Calcium then influxes into the neuron from the extracellular fluid moving into the cell. The calcium influx is going to cause those synaptic vesicles to fuse with the cell membrane. This releases acetylcholine into the synaptic cleft. Acetylcholine diffuses across the synaptic cleft and binds to receptors on the motor end plate of the muscle fiber. Now, these receptors on the muscle fiber are ligand-gated ion channels. So when acetylcholine, the ligand, binds to these receptors, they open up and sodium ions rush into the muscle cell from the extracellular fluid. At rest, a muscle cell's membrane is very negative, about -90 millivolts. We call this its resting membrane potential. When sodium, a positive ion, rushes in, it moves the cell membrane to become more positive, which triggers an action potential. The signal, or action potential, then travels along the sarcolemma into the T-tubules. This change in membrane potential along the T-tubules causes voltage-gated calcium channels in the sarcoplasmic reticulum to open, signaling calcium release channels to also open. This influx of calcium from the sarcoplasmic reticulum into the sarcoplasm floods the thin and thick filaments, allowing a muscle contraction to happen as we outlined in the last video. The effects of acetylcholine are brief because it is almost immediately broken down by an enzyme called acetylcholine esterase. This enzyme is found in the synaptic cleft on the extracellular side of the motor end plate membrane. Acetylcholine esterase breaks down acetylcholine into acetyl and choline, which can no longer activate the acetylcholine receptor. This entire sequence happens in just milliseconds. If there is another impulse, then more acetylcholine will be released into the synaptic cleft, allowing for another muscle contraction to happen. Muscle contractions will stop when the neuron is no longer releasing acetylcholine. Remember from the last video that once that action potential travels down the T-tubules, it triggers calcium release from the sarcoplasmic reticulum. That calcium is what lets myosin grab onto actin, starting the sliding filament mechanism. So the neuromuscular junction is really just the gatekeeper. It's where electrical signals from neurons are first handed off to muscle fibers so that contraction can happen at all. Let's bring this to life with a few real-world examples. Botulinum toxin, or Botox, is a toxin that blocks the release of acetylcholine from the motor neuron. Without acetylcholine, the muscle never gets the message to contract, leading to paralysis. Curare is a plant-derived poison originally used by South American indigenous tribes as an arrow poisoning. It binds to acetylcholine receptors, blocking acetylcholine from activating them. This also causes skeletal muscle paralysis. Myasthenia gravis is an autoimmune disorder where the body attacks its own acetylcholine receptors, making communication between neurons and muscles weaker. Patients often experience muscle weakness that worsens with activity. These conditions highlight just how precise and essential the neuromuscular junction is. Any disruption to its communication can have profound effects. So to summarize, the neuromuscular junction is the communication point between a motor neuron and a skeletal muscle fiber. A nerve impulse triggers the release of acetylcholine, which binds to receptors on the muscle fiber. This sets off a new action potential in the muscle, leading to calcium release and ultimately contraction. This system is tightly regulated so muscles contract only when they're supposed to and with the right amount of force. The neuromuscular junction is the critical bridge between thought and movement. Without it, the sliding filament mechanism could never even begin. In the next and final video of this muscle series, we'll dive into muscle metabolism, how your muscle cells actually generate and use energy to fuel contraction, from ATP to creatine phosphate, and from anaerobic glycolysis to aerobic respiration. We'll explore the different ways your muscles power movement. If you're new here, my channel is all about making biology, anatomy, and physiology clear and approachable for students and curious learners alike. So if you want to keep learning about how your body works, don't forget to subscribe and turn on notifications so that you never miss out on new content. Thanks for watching, and I'll see you in the next video.